It's my first visit to India ever, so I'm very impressed. And I took the risk and came all the way from UK. So thank you very much. May I ask first, how many people here are in the medical or biology field? Okay, quite a few. Because just to choose the language I will use, how simple or how jargon free will it be. So first, I also didn't know how long I thought it was about. How do you? At the National Cancer Institute in USA, and then after that I moved to Sweden, in Karolinska Institute for further, I had a grant for further studies, and then I went back to the United States, where I worked at the University of Arizona, College of Medicine, Phoenix, and then now I landed in the UK, and I don't know where next. So, yeah. So, I'm just going to talk about 3 topics. So, my career in cancer is... The overarching aim or goal of my studies is to try to understand cancer simply. Cancer biology. What is cancer? How does it initiate and progress? And how can we uncover or discover new targets inside the cell so that we can develop targeted therapies? So I work inside the cell. I'm not a physician, so I don't do the organs, the human. I work inside the cell. So I will focus on 3 topics. In the cell, there is something called signaling, which transfers the signal from outside of the cell to the inside of the cell. This slide might look a little bit complicated, but I will try to simplify it. So that's the outside of the cell. This blue line is the outside of the cell. And this is the nucleus, the inside of the cell. And this is the receptor that I was talking about, and how it transfers the signals from outside to the inside, leading to cell survival, proliferation, and avoiding of cell death. And this is how, not how, but one of the mechanisms that the cancer cell tries to survive, is to avoid dying, is to try to maintain its proliferation activities that actually was discovered long time ago, maybe in the 60s or 70s, but until now, in the 2026, we're still discovering new proteins that work with this receptor to increase cancer. Yeah. So in our field or in any scientific field, it doesn't stop. There is always everyday new discoveries. So we have identified, using certain technique called mass spectrometry, we could identify novel proteins that were not discovered before that work with this receptor. For example, the so-called PCNA, or this one, it acts as a protein complex manager, if you will, that will target certain processes in the DNA replication and DNA damage pathway. So the cells, when it experience damage, the DNA could do one of 2 things. Either it gets repaired automatically in the cells, or the cells die, or the cells could bypass this problem and do the so-called DNA damage tolerance, that I'll close an eye on the damage so that it just passes, and then I'll repair it later. So that's what we were discovered, that we have identified the mechanism by which our receptor bind to the replication protein and control the DNA damage pathway. And how this happens, this is structural model of our receptor and of the new protein, how they bind together, and the receptor, in our field, when you add a phosphate group, it activates the protein. So our receptor activates the PCNA on 3 sites, and this is how it regulates the DNA. Metastasis means when the cancer moves from one place to the other, to the other secondary place inside the body. So this protein existed, but we didn't know how it is regulated. So our receptor also binds to this protein and phosphorylates it, and causes or plays a role in metastasis of cancer as well. We did this in 2 types of cancer. Okay, so that was our second major discovery. The third topic I'm going to talk about is in pediatric acute myeloid leukemia, and that was done in America, University of Arizona. We worked on children cancer, acute myeloid leukemia, and what we have done is, leukemias, especially if it does not, it's resistant to chemotherapy, like we call it refractory, or relapsed when it comes back, okay? So what we try to do there, it's also very expensive to treat the refractory and relapsed leukemia. So they're exposed to drugs, the leukemia cells, in this environment compared to the regular screening environment, and see how this will alter the response, the leukemia cells' response to drugs. So this is just to show you the workflow. There's no need to show all this, but this is just showing on the left-hand side about 50 different drugs. And these are about 12, 13 different leukemia cell lines. And the blue and the red shows live cells, which means that the cells that have a red square did not die. The cells that have blue square, they died.